Recording.
Okay, I give you the word now. Please go ahead.
Okay, thank you, Ilse. Good afternoon, everybody. I am Rocío, and as Ilse mentioned, I'm a postdoctoral researcher working currently with her in our group in the topic of cartilage mechanobiology. I come from Spain. I arrived here to Leuven almost half a year ago, and I did my PhD in osteoarthritis, mainly working with stem cells. So today, in this class on mechanobiology, we are going to discuss a bit, as Ilse explained before, the mechanobiology of cartilage, and then Judith will explain the part of mechanobiology bone. So here in this slide, you can see an overview of today's class. So the first part would be mainly focused on cartilage mechano-adaptation, and the second part on bone mechano-adaptation and the bone-cartilage interface, and a little bit of insights into the osteoarthritis disease. As you can see here, we have divided each part of the class in a similar way or with a similar structure. So first, we are going to just refresh a little bit things that we think that you may already know from your previous studies about the structure and the function of both cartilage and bone. Then we will explain how are they designed to be able to adapt to loading. And finally, we will explain a little bit more in depth the mechanotransduction process. So let's start first with the part on cartilage mechanoadaptation. And I have put here in this slide my email. In case you have any question or any doubt after the class and you want to email me, I will be happy to try to answer all your questions. So first, a couple of general aspects about cartilage. So it is a type of connective tissue that is found at different locations within our body. So specifically, it's going to be present in those places that require a high degree of flexibility. So for example, we are going to find cartilage in our ears, in our spinal discs, or in our joints, such as the hip joint. Cartilage is composed mainly by cells, which are the chondrocytes, and the extracellular matrix, which is synthesized or produced by these chondrocytes. And the extracellular matrix is mainly formed or composed by water and different proteins, such as collagen and proteoglycans. So depending on the type of proteins found in cartilage and depending on the amount of each type of the proteins, we can differentiate three different types of cartilage. We have hyaline cartilage, elastic cartilage, and fibrocartilage. Hyaline cartilage is the most common type of cartilage in our body, and it's mainly composed of type 2 collagen fibers. So for example, we can find this type of cartilage in the synovial joints, for example, in our knees, the ankle, elbow, the hip, etc. Then elastic cartilage is the second type of cartilage in the body, and it's mainly composed of elastin fibers. And these elastin fibers are going to allow this type of cartilage to be very, very flexible. So this elastic cartilage can be found, for instance, in our ears. So if you touch your ear and you want to fold it a bit, you can do it because it's composed of elastic cartilage. And then the third type of cartilage in our body is the fibrocartilage, and it's characterized by the presence of type 1 and 2 collagen fibers. So location of this type of cartilage or an example of this cartilage can be found in the intervertebral discs. So for this class, we are going to mainly focus on hyaline cartilage, which is also known as articular cartilage. And as I introduced a bit earlier, this articular cartilage is mainly present in our synovial joints. So the synovial joints are those joints that allow for a wide range of movement, and they are characterized by the presence of the joint cavity. So surrounding this joint cavity, we have this fibrous tissue, which is going to form the articular capsule. So inside this articular capsule, we have two bones that are articulating, and in the surface of the bones that are articulating, we can find the articular. So the function of articular cartilage in that place, so covering the end surface of the bones, is on one hand to allow for distributing the joint contact forces over a large area in order to minimize the peak loads that are going to be transmitted to the subchondral bone. And on the other hand, articular cartilage, it's also going to allow to minimize the friction to wear. So this way, the articulating bones can move smoothly against each other without damaging the subchondral bone or the underlying tissue. For example, in this slide, I have included this table, which I really like because you can see the coefficient of friction of different materials. So for example, if you have a look to the coefficient of friction of ice on ice, which of course is a very slippery material, this coefficient of friction is 0.1. Also, if we have a look to this material, which is the one that is used for the artificial joints, the coefficient of friction is around 0.01 or 0.05. So then in table 3, you can see the coefficient of friction of the native articular cartilage, and you can see that it's around 0.005. So it's much lower than any material that we are able to create. So as human beings, we haven't been able to create a material that is better than our native articular cartilage, which I think is very impressive. So then the other ability or the other function of articular cartilage is to minimize the contact pressures that are going to be transmitted to the subchondral bone. So as you can imagine, during our daily locomotion or our daily lives, if we practice any type of sport, our spine, our lower limbs are going to be submitted to very high peak loads. So for example, I have put here several examples of the peak loads that we can find in the hip. When we are walking, for example, the hip has to withstand 6 times the body weight. And if we are jumping, the hip it's going to experience forces of 10 times our body weight. So how is it possible that with all this loading or with these impact forces, articular cartilage is not damaged and is still fine and healthy? So this is because the composition of the tissue is precisely structured or precisely composed in order to be able to withstand this loading. So if you remember from before, the articular cartilage has a cellular component, which are the chondrocytes, and has the extracellular matrix. And this extracellular matrix has water and different proteins, mainly type 2 collagen and proteoglycans. So it's going to be this composition of the extracellular matrix that is going to allow cartilage to support this mechanical loading. So if we, here we have cross-section of articular cartilage. So if we were able to do a zoom in this extracellular matrix, we could see something like we have here in this picture. So we have a network mainly composed of collagen fibers and proteoglycans. Proteoglycans such as the aggrecan that we have here highlighted. So what happens with the aggrecan or the proteoglycans is that they are negatively charged, and therefore they are going to absorb water, and therefore they are going to absorb the impact shocks. And then we have the collagen that is going to provide cartilage with its tensile strength. So due to this composition, it's going to be something similar as when we are in a car crash, for example, and we have this airbag. This is supposed to be a gift that is not working, but you could see here a car crash, and you could see the airbag coming out and absorbing the impact of this accident. So in the same way as if we are in a car crash, we will have this airbag that is going to absorb the impact forces. In cartilage, we have this extracellular matrix with these proteoglycans that are going to absorb the forces. So collagen in the extracellular matrix is going to provide cartilage with its tensile strength, support, and structure. And then we have the aggregate that helps to dissipate the impact forces. So, but how is this actually achieved? So how this matrix can absorb this or dissipate these impact forces? So we are going to explain this in the next slide. So here in the left side, you have, let's say, cartilage or the extracellular matrix of the cartilage in an initial situation. So in this situation, we have the aggregate, which has a lot of negative charges, and therefore it's going to bind very strongly to water molecules, because water molecules have positive charges. So therefore, in this, let's say, normal situation, we have the aggregate that is going to be surrounded or bind to a bunch of water molecules. So therefore, this aggregate is going to try to expand or to swell. On the other hand, we have the collagen forming also a network that is pretensed. And this collagen network is preventing the aggregate from expanding too much or from swelling too much, which is what we call the swelling pressure. So what happens if at this point articular cartilage experiences impact or a load? So let's say that I jump. So what happens when I jump? So when I jump, what happens is that articular cartilage is gonna experience a loading, and therefore when this load is higher or exceeds this swelling pressure of the aggregate, these water molecules that are surrounding the aggregate are gonna be released from this area. And therefore, since the aggregate is losing the water molecules, this protein is gonna it's gonna change, or it's gonna be deformed, and therefore the loading is gonna absorb. At the moment that this loading or this mechanical stimulus disappears, then the water is gonna go back or rush back to the aggregate because of few main reasons. So one is that water is gonna move in favor of the concentration gradient. So from a high concentration to a low concentration. And then because the aggregate, which has these negative charges, is going to attract the water molecules. So once this water goes back to this section of the extracellular matrix, the aggregate would recoup its initial shape, and therefore cartilage will recoup its initial shape. So this is compression and expulsion of the water, and when the water is captured again, it's what explains the ability of articular cartilage to support the loads. Is there any doubt at this point? Do you want me to repeat something or to clarify something? No? Okay, I guess that's a no, so we can continue. Okay, so this is what happens at the level of the extracellular matrix with the collagen and the aggregate. But if you remember well, in articular cartilage, we also have the cells. So we also have the chondrocytes. And when physical stimuli are applied to articular cartilage, the chondrocytes are going to deform. And also in the matrix is going to be changes in the hydrostatic pressure, in the fluid flow, the ion concentrations, etc. So here I have a question for you. So do you think that these physical stimuli, apart from changing or from deforming the chondrocytes, are going to have an impact on the biology of the chondrocytes? So for example, on which genes are going to be expressed or repressed? So the answer is yes. Yes, these physical stimuli are going to have an impact on the biology of the cells. So I'm going to explain now how this is going to happen in cartilage. So probably during your degree, you have been said that chondrocytes are somehow a very lazy cell. So I don't agree 100% with that. So it's not 100% true. It is if we compare the metabolism of the chondrocytes with other cell types, such as epithelial cells or another kind of cells. But the metabolism of chondrocytes is very, very important to ensure that cartilage keeps its homeostasis, so that cartilage stays healthy. And why? It's because the metabolism of the chondrocytes is going to ensure a balance between the synthesis of extracellular matrix and degradation of specific components. So this way, with this balance, the extracellular matrix is going to be always the optimal extracellular matrix to be able to support or to withstand the mechanical challenges during, for example, daily locomotion. So here you can be wondering, okay, but what is the exact relationship between this chondrocyte metabolism and mechanical loading? So the reason why I'm explaining this is because mechanical loading is known to affect the metabolism of the chondrocytes. So specific types of loading are going to induce biosynthesis of specific components of the extracellular matrix. It's going to help to keep this balance between catabolic and anabolic processes, so this balance between synthesis and degradation of the matrix. It's going to help to preserve the integrity of the tissue and therefore to maintain the mechanical properties. So in this slide, I have put two examples of how important mechanical loading is to keep our cartilage healthy. So for example, in this first paper or this first study, the authors report that rats that went for a month in a Russian spacecraft, once they were back, they showed early signs of cartilage degradation and early signs of osteoarthritis due to the ingravity. So because mechanical loading was not applied to these rats, and therefore the cartilage was degraded. And then in this second experiment, they kept the mice, like you can see here in this picture. So the legs in the back were not submitted to loading, and they observed a thinning of articular cartilage in these limbs because of the immobilization. So this shows that it's very clear that mechanical loading seems to be the trigger that it's necessary to turn on the cell machinery and therefore ensure that chondrocytes are going to keep this balance between synthesis and degradation of the matrix. That is going to ensure that cartilage is in a healthy situation, that homeostasis is maintained, and that this cartilage can of course, withstand and resist this impact and can be in a healthy status. So here, a question that you may have is, okay, how is it then possible that an external physical stimuli is going to produce a biological response? So it's going to produce that more extracellular matrix genes are expressed. So this is thanks to a process that we know as mechanotransduction. So mechanotransduction can be defined as the process whereby the cells are going to convert these physical mechanical stimuli into biochemical responses. So this process of mechanotransduction explains how physical stimuli are going to have an impact on the biology of the cells. So for example, if we have here this schematic representation of a chondrocyte, if we applied mechanical loading, this is going to cause the formation of the matrix, the formation of the cells, and also changes in the fluid flow, ion concentrations, etc. All these changes are going to be perceived or felt by different mechanosensors or receptors that are present in the membrane of the chondrocytes. And when they are activated, they are going to trigger specific intracellular responses that would finally lead to the expression or suppression of specific genes. And these genes are going to dictate if more matrix needs to be synthesized or if specific elements need to be degraded in order to adapt the extracellular matrix to the mechanical challenge. So this mechanotransduction process is normally divided in three different phases or three different stages. So the first one is mechanocoupling. The second one is biochemical coupling, which is also known in literature as mechanotransduction per se. And then we have the mechano-adaptation, sometimes known as effector response. So here in this scheme, again, you can see how these different phases occur during the whole process of mechanotransduction. So when we have a mechanical loading of joint cartilage, the first step is the mechanocoupling, where it's produced the transmission of forces from the extracellular matrix to the chondrocyte's surface. So the next step is the mechanotransduction. So these receptors in the surface of the chondrocytes are going to recognize these physical forces, and they are going to transform these forces into intracellular responses. And finally, the cells, or the metabolism of the cell, is going to adapt in order to remodel the articular cartilage or the extracellular matrix to be able to adapt to this new mechanical environment. So as you can imagine, this entire process of mechanotransduction with its different phases, it's key for ensuring that cartilage homeostasis is maintained. But not only these independent processes are required, but also different elements in each phase of the process need to be working in a perfect way to ensure that these mechanical stimuli are perceived in a correct way to help maintain cartilage homeostasis. So now I'm going to explain different elements that are very important in each one of these steps to have an effective mechanotransduction. So the first element which is very important in this case for the mechanocoupling phase, which is the first phase of the mechanotransduction process, is the extracellular matrix and the pericellular matrix. So as we explained before, the extracellular matrix is going to be able to absorb the impact forces thanks to the presence of collagen and the presence of proteoglycans. But apart from this function, the extracellular matrix is also very important to deliver mechanical signals to the chondrocyte. And to understand how this happens, you need to know that this extracellular matrix is divided into two major components. So one is the territorial matrix, which is this part of the matrix that is far away from the chondrocytes. And then we have the pericellular matrix that you can see here in this picture. So the pericellular matrix is a region of around 1 to 5 millimeters that is immediately surrounding the chondrocyte. So this part of the matrix is a bit different because instead of having collagen type 2, it is very rich in collagen type 6. And instead of having aggrecan, it's very rich in another proteoglycan, which is the perlecan. So this pericellular matrix has key mechanosensing properties, and it's known to be able to amplify the mechanical signals that are transmitted through the extracellular matrix to the chondrocytes. So this transmission of forces from the extracellular matrix to the chondrocytes can happen in two different ways. So one way of doing this is by mediating the direct contact of the pericellular matrix with different mechanosensors that are present in the membrane of the chondrocytes. And the other way of transmitting these forces is because the pericellular matrix acts as a repository of different molecules and growth factors. So these molecules are going to be stored in this area, so in this pericellular matrix, maybe being bound to perlecan or in the middle of the network. And once mechanical loading is applied, these molecules are going to be released, and they would trigger also specific responses in the chondrocytes. So this is the mechanisms that are currently known on about how this pericellular matrix contributes to the process of mechanotransduction, and more specifically to the process of mechanocoupling. So then the next step, or the next phase in the process of mechanotransduction, is the biomechanical coupling. So in this process is initiated by stimulating different mechanoreceptors or mechanosensors that are located in the membrane of the chondrocytes, as I already mentioned in the previous slide. So once these mechanosensors that are present in the membrane of the cells are activated, then they are going to trigger specific intracellular responses in the chondrocytes. So currently in the literature, there are a lot of different mechanosensors or candidates to be mechanosensors in chondrocytes. But for this class, we have just chosen the three main ones that are known to be actually playing a role in this mechanotransduction process. So these mechanoreceptors that participate in the mechanotransduction of chondrocytes are ion channels, integrin receptors, and primary cilia. But you also have to bear in mind that there are also more protein-specific receptors present in the membrane of the cells that are going to trigger also specific pathways or signaling pathways when mechanical loading is perceived. But for this class, we are going to focus mainly on these three types of mechanoreceptors. So first, the ion channels. Normally, there are many types of different ion channels, but in the case of chondrocytes mechanotransduction, the calcium channels are the most important ones because intracellular changes in calcium concentrations are one of the most fundamental mechanical responses to mechanical loading. So there are in chondrocytes mainly two types of channels that are going to regulate the calcium influx in response to loading. So one of them are the piezo channels and the other ones are the TRPV channels. So in the case of the piezo channels, they are going to respond to high levels of mechanical stress. So therefore, they are going to be activated when strains of around 13 to 45% are applied. And therefore, they are going to be related with processes of cell death and apoptosis. So for instance, when a non-physiologic mechanical stimulus arrives to the cell, these channels are going to be opened and a lot of calcium is going to come into the cells to such a level that it's going to be toxic for the cell and it's going to cause cell death and apoptosis. So therefore, this is a type of channel that is going to be related with cartilage damage. And then we have the second type of channels, which are the TRPV channels. And they are, on the contrary, what happened with piezo channels, they are going to respond to physiologic or physiological mechanical stimulation. So they are going to be activated with strains of around 3 to 8%. Since this type of channels are more related to more physiologic stimulation, the loss of these channels have been proven to be associated with the disruption of the homeostasis of the cartilage and therefore with having a role in the osteoarthritis development. So there are more types of ion channels, but these two ones are known currently to be playing a very, very important role in the mechanotransduction process in chondrocytes. So another type of receptor that plays a role in mechanotransduction in chondrocytes are the integrin receptors. So the integrins are proteins that or transmembrane proteins that are going to link the extracellular space with the intracellular space. And therefore, they are sometimes referred to as the molecular bridge between the PCM or the pericellular matrix and the intracellular space. So as you can imagine, when a mechanical stimuli arrives from the extracellular matrix, it's going to be sensed or perceived by this domain of the integrins, which are going to then activate intracellular signaling cascades that are going to mediate a cellular response. So these integrins are known to be playing a very important role on mediating the synthesis of new extracellular matrix components. So this is another type of mechanosensor or mechanoreceptor that is playing an important role on keeping cartilage homeostasis through the process of mechanotransduction. And then the third type of mechanosensor that we are going to explain today are the primary cilia. So these cilia are structures that appear in the membrane of the chondrocytes that are going to protrude to the pericellular matrix. So in this picture, you can see here a scheme of primary cilia, and here you have a real picture of how a primary cilia looks like. So this is a picture taken with transmission electron microscopy, in which you can see a chondrocyte, and here you can see the primary cilia. Here it's the same, but with a higher magnification, so you can see the structure of the primary cilia. So these primary cilia are sensitive to mechanical changes, and in fact, they have been proven to change the length or their length in response to mechanical stimuli. However, in the case of the primary cilia, they are not going to be the primary mechanoreceptor, but instead they are going to activate different downstream pathways that are going to activate or repress a specific response as a result of the mechanical loading. And one example of a regulator that it's going to be linked to this primary cilia is the transcription regulator cited 2. So this is a protein that it's going to mediate the process of mechanotransduction by means of the primary cilia. So at this point, we have seen 3 types of mechanoreceptors that are present in the membrane of the chondrocyte. So we have seen the ion channels, the integrin receptors, and the primary cilia. So once these receptors are activated, then different intracellular mechanosignaling pathways are going to be also activated in response to this stimulus. So in this slide, I have included several examples of the pathways that are known to be more related to the mechanotransduction process. So for example, we are going to have changes in the cytoskeletal elements. Another pathway that is activated in response to loading is the MAP-ERK pathway, the Wnt pathway, TGF-beta SMAD, HippoYAP, or also the microRNAs. So you don't need to know, of course, all these pathways or the details of these pathways, but I would like you to remember that after this mechanotransduction process, these pathways are going to activate or to lead to specific gene expression in the cell. So these pathways are going to activate or repress specific genes in the nucleus of the cell. So then we will be in the last step of the mechanotransduction process, which is the mechano-adaptation or effector response. So the activation or the suppression of these cascades that I mentioned before are going to regulate the cellular functions. So are going to decide if the chondrocytes need to produce more matrix or to create some parts of the matrix so that this matrix can be optimized to withstand the mechanical loading that it's perceiving in a specific moment. That would be the mechano-adaptation and the last part of the mechanotransduction process. So the response or the final response of the chondrocytes to this mechanical loading, it's going to depend on different things. So for example, the type of loading, the magnitude of the loading, the frequency, the loading history, all these parameters are going to have an impact on how the chondrocyte responds to this stimuli. So still there's a bit of controversy on which type of stimuli are going to produce which type of response in the chondrocyte. But at the moment, it's more or less clear that dynamic low magnitude loading, so a more physiologic mechanical loading, it's going to lead to increased synthesis of the extracellular matrix components, and it's going to inhibit also inflammatory pathways. So you don't need to know this, of course, but it's just to show you that the different parts of the mechanotransduction process that we've just seen are going to lead to the suppression of inflammatory gene expression, for example, and finally are going to lead to matrix synthesis. However, on the contrary, if we have a non-physiological loading, either overloading or reduced loading, as we saw with the mice that were to space in a spacecraft. So non-physiologic loading, it's going to produce a decreased expression of the genes that are going to lead to extracellular matrix synthesis, and therefore it's going to lead to cartilage damage. So just to finish my part, as a summary or important concepts that I would like you to remember after this first part of the class is that the composition of articular cartilage is perfectly designed to support mechanical loading and also to provide frictionless movement. And this is going to be thanks to the presence of the proteoglycans that are going to absorb water, and also because of the presence of collagen type 2. Then we have seen that biomechanical factors are going to play an important role in the health or in the homeostasis of synovial joints, because chondrocytes are going to perceive these physical signals from the environment using a wide variety of mechanisms. So in this class, we have had a look to the ion channels, the integrin receptors, and also to the primary cilia. So the process by which these physical stimuli are going to be converted into biochemical response is the process that we know as mechanotransduction. And it's going to explain how the forces are going to have an impact on the biological response of the cells. In general, by means of this mechanotransduction process, moderate mechanical loading is going to induce different signaling cascades that would lead to the production of extracellular matrix components. However, if we have a non-physiologic loading, either overloading or reduced loading, this is going to lead to cartilage damage and, for example, the development of specific joint diseases such as osteoarthritis. So I don't know if you have any doubt at this point or anything that you want me to clarify.
Thank you, Rocío, for the very nice overview of the cartilage adaptation and mechanobiological factors that play a role in cartilage health. Are there any questions here? I see no questions from the audience. Maybe I just want to add that in our research group, we are looking into these processes by using different models. So looking at the human joints and looking how the cartilage adapts there to the loading that we calculate with the modeling. We're also using some animal models, which Judith can also talk about. That's the part of her research. And you are mainly using our bioreactor setup to look at how cartilage tissue and cells that we load are changing their expression as a result of the loading. Mm-hmm. No questions in the meantime from the students?
Everything was clear.
I think everything was clear. Okay. Then maybe I would like to suggest that we take a small break here because I can see that we're a bit earlier, but maybe let's take a 10-minute break and be back at 14:20, and then we can go to the bone mechano-adaptation part of the class. Any objections by the students? Okay, I hope you're still alive. Otherwise, we are just doing this for Natty class. Perfect. Okay, good. See you all at 14:20. Okay?
Perfect.
Thank you.
Thank you.